Hello everyone, today we're going to talk about glycolysis regulation. In the previous video, we talked about glycolysis and how it occurs. But today, in this video, we specifically want to talk about how our body can regulate this pathway. Because the regulation of different pathways are so important for us. So let's get started with glucose. This is our glycolysis, right? In glycolysis, we had two steps. In the first steps, we used two molecules of ATP, and in the second phase, we produce four molecules of ATP. And also, we produce two molecule NADH in the phase 2. Now, if you pay attention to these arrows, some of them are black. This one, and this one. These are important reactions and important enzymes that can be regulated by different hormones or molecules. Totally, we have two types of regulation. We have two types of regulation. It can be hormonal and it can be allosteric. Hormonal means we can add phosphate groups or remove phosphate groups from an enzyme. And in allosteric, it means that we can add ions and molecules in an enzyme or remove them. So let's get started with the first enzyme here. The first enzyme here in this reaction, glucose to glucose 6-phosphate, is hexokinase. Hexokinase is the key regulatory enzyme in this reaction. So let's think why we should increase the speed of glycolysis. Glycolysis is a catabolic pathway because at the end, because we break a big molecule into a smaller one and we produce energy at the end. It's a catabolic reaction. In this step, we turn glucose into glucose 6-phosphate. So, when we have a lot of glucose in our cells, when we have a plenty of glucose in our cells, it is logical that the cell wants to turn glucose into something else. And what can it be? It can be glucose 6-phosphate. So glucose allosterically can enhance and stimulate hexokinase. Let's talk about it in another way. If we have a lot of amount of glucose 6-phosphate, so is it necessary to turn glucose into glucose 6-phosphate? No, because we already have a lot of them. So glucose 6-phosphate can be a inhibitor. It can allosterically inhibit hexokinase. Let's talk about hormonal regulation. We have insulin. What insulin do? The main role of insulin is to enter the glucose into the cells. For example, if we have vessels here, insulin helps glucose to import them actually into the cell. So when insulin import glucose into the cell, it means that glucose increase. So it doesn't matter if we say our insulin is increased or glucose is increased. Both of them can enhance and stimulate hexokinase because insulin wants to enter glucose into the cell. So more glucose, more glycolysis, and it can enhance and stimulate hexokinase. In contrast of insulin, we have its enemy. What's its enemy? The hormone that can act completely in different direction. It is glucagon. Glucagon wants to exit glucose out of the cell. So glucagon can hormonally actually inhibit hexokinase. Let's talk about next reaction and the next important key regulatory enzyme. It is phosphofructokinase 1. Phosphofructokinase 1. It turns fructose 6-phosphate into fructose 1 and 6-bisphosphate. So, how it can be regulated? For the first allosteric molecule, We can say ATP is an important regulatory in most enzymes. How can ATP regulate this molecule or even another enzymes, this enzyme and another enzymes? Let's think logically. Why we are doing glycolysis? To produce ATP, right? To produce ATP, to produce energy. So let's think our body has a lot of amount of ATP, has a lot of amount of energy. Do we really need to produce more ATP for our body? We already have a lot of them. So no. So in this case, phosphofructokinase 1 will be inhibited by ATP. Our body has a lot of ATP. We don't need any more ATP. So when phosphofructokinase 1 senses that our body has a lot of amount of ATP, it says that stop phosphofructokinase 1. We don't want ATP. We don't want to continue glycolysis, and it inhibits PFK1. In another way, we have another molecule called citrate. What is citrate? Citrate is it's the first molecule that can be created in Krebs cycle. Let me tell you something totally in all regulatory pathways. For example, in this enzyme, some reactions are before PFK1 and some of them are after PFK1. For example, we have turning glucose to glucose 6-phosphate. It is before PFK1. And for example, PEP to pyruvate, it is after PFK1. We can say most of the times, 99% of the times, the molecules which are after the enzyme, after our key regulatory enzyme, those molecules will inhibit the enzyme. And whenever we watch the molecules before the enzymes, for example, glucose 6-phosphate, these molecules will stimulate the enzyme. In other words, everything we have from here backward will be stimulatory, and everything we have here and in continue will be inhibitor. And when our pyruvates go to aerobic pathway, aerobic system, pyruvate turns into acetyl coenzyme A, and acetyl coenzyme A will turn into citrate, which is in Krebs cycle. So citrate is a inhibitor for PFK1. We talked about ATP. We talked about when our body has a lot of energy. But let's talk about when we don't have enough energy, when we are tired or when we want to produce energy. For example, we're doing some physical activities, we're running, we're climbing, and our body requires to start and increase the speed of glycolysis. In the case of ATP, and when our body do not have enough energy, we don't say our ATP decreased. We say our ADP increased. We know that ATP turns into ADP and inorganic phosphate when our body wants to use ATP to produce energy. So when we turn ATP a lot to ADP increase. And the increase of ADP in our cells, in our body, means that our body requires energy and it is tired. We should follow pathway, we should stimulate pathway to produce more energy. What glycolysis do is produce energy. So the increase of ADP in our cells increase and stimulate PFK1 activity. So let's talk about this complex. This part is so important because it might be a little complicated. We have this enzyme complex. What this enzyme does? We have fructose 6-phosphate. We have fructose 6-phosphate here. This complex This complex has two enzymes, PFK2, phosphofructokinase 2, and fructose 2 and 6 bisphosphatase. In the first step, we're talking about PFK2. Fructose 6 phosphate, which comes from here, can turn to something we call fructose 2 and 6 bisphosphate. Fructose 6 phosphate turns into fructose 2 and 6 bisphosphate because this is a kinase enzyme. Kinase enzyme add another phosphate group to molecules, and fructose 6 phosphate turns into fructose 2 and 6 bisphosphate. The fun fact is fructose 2 and 6 bisphosphate can stimulate PFK1. In other words, fructose 6 phosphate can indirectly stimulate PFK1. How? By turning into fructose 2 and 6 bisphosphate using PFK2 and 2 and F2 6 bisphosphate, fructose 2 and 6 bisphosphate stimulates PFK1. But we cannot keep fructose 2 and 6 bisphosphate in our body forever and stimulate and continue to stimulate PFK1 forever. So we have to do something to get rid out of this molecule. What we do? We have this enzyme here, fructose 2 and 6 bisphosphatase. Phosphatase. What phosphatase enzymes do? They remove phosphate group. So let's remove a phosphate group from this molecule, and it again turn into fructose 6 phosphate. So we can say PFK2 can actually stimulate PFK1, and fructose 2 and 6 bisphosphatase can inhibit PFK1. Hormonally, we have two hormones, right? Insulin and glucagon. Insulin can do his act on both of these enzymes. It can add phosphate to both of these enzymes. When PFK2 receives these phosphate, it will be inhibited. When glucagon acts on PFK2 and when PFK2 receives phosphate groups, it will be inhibited. So we can say glucagon can inhibit PFK1, because whenever we inhibit PFK2, it means that we cannot turn fructose 6 phosphate into fructose 2 and 6 bisphosphate anymore, and it cannot stimulate PFK1 anymore. So glucagon can inhibit PFK2 and inhibit PFK1, but it can stimulate fructose 2 and 6 bisphosphatase. So when it stimulates this enzyme, when it stimulates fructose 2 and 6 bisphosphate, it helps this enzyme to turn F2 6 bisphosphate into fructose 6 phosphate, and it's also help to inhibit PFK1. So totally we can say glucagon can hormonally inhibit PFK1. Now let's talk about insulin. What insulin do? Insulin can remove these phosphate groups. Glucagon can add phosphate groups. Insulin can remove these phosphate groups. So whenever we remove phosphate groups from this enzyme PFK2, it will be stimulated and it can turn F6 phosphate into F26 bisphosphate, and it will stimulate PFK1. So insulin, we can say, have a stimulatory action on PFK1, and also it can inhibit fructose 2 and 6 bisphosphatase. When this enzyme inhibited, it means that we have less we have less fructose 6-phosphate, we have less this reaction, fructose 2 and 6-bisphosphate to fructose 6-phosphate, and it stimulates PFK1. So totally, we can say insulin can stimulate PFK1. Now, let's talk about our final enzyme, pyruvate kinase. The first molecule that we can talk about is ATP, adenosine triphosphate. We talked about it before. How can ATP totally regulate glycolysis? So we do glycolysis because, in our muscle cells especially, because we require energy. So when we have a lot of amount of energy, do we need to enhance and stimulate glycolysis? No. When we have a lot of ATP, we don't want glycolysis to be continued. So ATP can actually inhibit pyruvate kinase. We have another molecule called long-chain fatty acids. Long-chain fatty acids can inhibit pyruvate kinase. Why? For example, whenever we're running for example, for 5 kilometers, our body prefer to use fatty acids to produce energy, and our body don't want to use its glucose anymore. Glucose is important for us, but fatty acids are only used to produce energy when we do some activities. So whenever we have a lot of long-chain fatty acids, it means that we do not require to use glucose anymore to produce energy. So long-chain fatty acids says to pyruvate kinase that I'm trying to produce energy. We don't need you anymore. So stop continuing glycolysis. So totally long-chain fatty acids can inhibit pyruvate kinase. We have another molecule called acetyl coenzyme A. Acetyl coenzyme A, as we said before, whenever we are in aerobic condition, our cells are in aerobic condition, pyruvate can turn into another molecule, which is A-CoA. So A-CoA is the forward, is after pyruvate kinase, and when we have a lot of acetyl coenzyme A, it means that we have a lot of A-CoA in our cells, and we don't want to produce more because we already in our aerobic condition. Our body, our cell is going to produce a lot of amount of energy from aerobic system. We don't need any more to produce pyruvate to turn it into acetyl coenzyme A. So A-CoA can inhibit this enzyme. Glucagon, our lovely hormone, just like before we talked about here, can inhibit this enzyme because glucagon wants to actually exports glucose out of the cell, and it don't like to continue to glycolysis. Whenever we have glucagon, we have insulin, and in contrast of glucagon, insulin can enhance and stimulate pyruvate kinase. And we have fructose 1 and 6-bisphosphate. This molecule specifically can directly, this is important if you have any exam, this is very important, that F1,6-bisphosphate can directly, not indirectly, can directly stimulate pyruvate kinase. And it can say to pyruvate kinase, please turn PEP or phosphoenol pyruvate into pyruvate, and we want to continue glycolysis. Now we already know how glycolysis is regulated by three main enzymes: hexokinase, phosphofructokinase 1, and pyruvate kinase. And we know how this enzyme complex can use fructose 6-phosphate to indirectly regulate this reaction and regulate phosphofructokinase 1. I really hope you enjoyed this video. Please push the like button and comment on the comment section. Your comments are really important for me. And as always, sorry if I'm not fluent enough to talk about these reactions in English. I'll try my best in every video to actually present better and better. And I really hope you enjoy this video. See you next time.